Hello everyone, I'm Rakesh, CEO and founder of Celerite Labs, and today I'm going to tell you how we are improving the process of cancer drug development and discovery. Cancer drug development and discovery is a long and expensive process. Preclinical testing of these drugs or therapies takes around 2 to 4 years and has a low success rate of around 31%. Current methods used in the preclinical testing does not mimic human physiology. One of the alternate methods is to form these 3D cancer tumors outside the human body and screen these drugs. However, the method takes 3 to 4 days to form these tumors. They're complicated as it requires multiple steps, and often the tumors formed are not reproducible. The lab research scientists, research institutions, and pharmaceutical organizations involved in this routine testing are mostly affected by this. We at Celerite Labs have developed a magnetic self-printing technology that is non-invasive, scaffold-free, and allows the formation of cancer tumors within 3 hours. Our method is 10 times faster than the existing methods, produces reproducible spheroids that mimic human physiology, and is also 50% cost-effective. The method is also compatible with the existing high-throughput screening systems. The global 3D cell culture market is of $845 million and is growing annually at 10.5%. Market share of the 3D spheroids and organoids is 42%, which is the serviceable available market. Our initial target customers are the research laboratories and institutes that comprises 23% of the serviceable available market and is $82 million. This market comprises of around 4,100 research labs and institutions and 300 companies that are developing different therapeutics within North America. During our customer discovery, we learned that the reproducibility of these tumors and physiological relevance is very important. We at Celerite Labs are filling this gap with our magnetic cell printing technology.